Associate Professor Dr. Shawit Ketgao, Associate Dean for Strategy, Research and International Affairs. Together with Mr. Benoit Nadeau, Assistant Dean for Special Affairs and International Collaboration from Khon Kaen University International College.
It works. Good afternoon, everyone.
กราบเรียนดร.ณรงค์ชัย อัครเสรณี นายกสภามหาวิทยาลัยขอนแก่น กรรมการสภามหาวิทยาลัยขอนแก่น รองศาสตราจารย์นายแพทย์ชาญชัยพานทองวิริยกุล อธิการบดีมหาวิทยาลัยขอนแก่น Mr. Uwe Morawetz, the founding chairman of the International Peace Foundation, Professor Randy Schekman, the 2013 Nobel Prize Laureate in Physiology and Medicine, our today's speaker. Honorary Council of France at Khon Kaen, other distinguished guests, ladies and gentlemen, my name
Enjoy the presentation.
Benoit and I are here to guide you through the conference, an event that is brought to you under the banner of Thai-Japan-ASEAN Bridges series. Today, the Bridge series looks at the role of public universities as an engine for social change.
Actually, in fact, Ajahn Chavez and I both believe that this topic truly resonates with us, both on our academic life, our professional life, as well as our personal lives. And KKU is truly honored to be able to host this event. And we also would like to take this opportunity to thank all those who have made this event possible. Of our society. The topic of academic accessibility is very relevant in a world where the cost of education continues on growing. And public universities play a great part in trying to provide higher education for all, at least for all who desire it. And this is more crucial than ever.
That's right. Our young graduates need to be prepared for the world of tomorrow. And universities can help in providing them with the skills needed to meet the challenges of a sustainable society.
Correct. Public universities also have been here for a long time, but they remain a key player in our economic, social, and cultural and political development. And this is true here in Thailand, but this is true all over Asia and, in fact, all over the world. In fact, higher education has never been more international than it is now, and higher education will continue to be a global force for growth, change, integration, and cooperation.
That's right. Now, Benoit, enough about us.
Yeah.
It is time to turn this wonderful event over our dignitaries and guest speakers. I would like to first start by extending our gratitude to our sponsors. Of course, Khon Kaen University, Khon Kaen University International College, College of Local Administration, Faculty of Humanities and Social Sciences, Faculty of Medicine, Faculty of Science, and of course, the Thai-Japan-ASEAN Bridges series. ผมขออนุญาตกราบเรียนเชิญรองศาสตราจารย์นายแพทย์ชาญชัยพานทองวิริยกุล อธิการบดีมหาวิทยาลัยขอนแก่นขึ้นกล่าวต้อนรับแขกผู้มีเกียรติทุกท่านที่มาร่วมงานเสวนาวิชาการในครั้งนี้กราบเรียนเชิญครับ And now please join me in welcoming Associate Professor Chanchai Pantong Viryagun, MD, and the President of Khon Kaen University.
Chairman of Khon Kaen University Council, Professor Schekman, Mr. Morawetz, founding chairman of the International Peace Foundation, Khon Kaen University Council members, ladies and gentlemen, I am delighted to welcome our distinguished guests, esteemed colleagues, and students to the Thai-Japan-ASEAN Bridges series of the Nobel Prize Dialogue, happening right here at Khon Kaen University this afternoon. Today's event holds special significance as we celebrate the 60th anniversary of our university. Together, we are here to explore the meaningful topic of the role of public universities as an engine for social change. A dialogue inspired by the profound insight of Nobel Laureate in Medicine, Professor Randy W. Schekman. Over the past 60 years, Khon Kaen University has made remarkable progress in achieving academic excellence while fulfilling our social responsibilities. We have played a crucial role in driving positive changes in society, guiding our communities and reaching beyond national borders to promote global cooperation for mutual development. It's noteworthy how Professor Schekman's scientific achievements and commitment to expanding knowledge closely align with Khon Kaen University's core value, rooted in social engagement. On this significant occasion, I eagerly look forward to the enriching discussion ahead. I would like to express my sincere appreciation to Professor Schekman for graciously sharing his invaluable insights with us. I also extend my gratitude to the International Peace Foundation, led by Mr. Morawetz. I am grateful to all of you for participating in this event. Your presence truly enhanced our dialogues and contributed to our collective pursuit of global development. Thank you very much.
Thank you. Thank you, Mr. President, for your welcoming comments. Truly appreciate it.
ลำดับต่อไปขออนุญาตกราบเรียนเชิญดร.ณรงค์ชัยอัครเสรณีนายกสภามหาวิทยาลัยขอนแก่นขึ้นกล่าวเปิดงานขอกราบเรียนเชิญครับ Now I would like to invite the chairman of the University Council, Dr. Narongchai Akara Serani, to address the opening of this event.
Thank you for the invitation. Good afternoon, ladies and gentlemen. Let me address Professor Schekman and also Mr. Uwe Morawetz, who is the founding chairman of the International Peace Foundation, and Khon Kaen University Council members, ladies and gentlemen. It's a privilege to stand before you today at the outset of what promises to be a thought-provoking and inspiring dialogue on the role of public universities as an engine for social change, the subject of our great interest at Khon Kaen. So on behalf of Khon Kaen University Council, I warmly welcome all of you to this Nobel Prize Dialogue, an event that symbolizes the bridge of knowledge and collaboration between Thailand, Japan, ASEAN, and beyond. The role of public universities in our society has never been more crucial. As institutions entrusted with the task of providing higher education, we are responsible for leading the change towards a more prosperous and equitable future for all. Our role is to innovate, educate, and serve as engines of social change. We embrace this responsibility with modesty and determination. The presence of Professor Randy Schekman among us today emphasizes the significance of our gathering. His groundbreaking work and dedication to advancing our understanding of the life science exemplify the transformative power of academic excellences. As some of us know, Professor Schekman worked at University of California at Berkeley, where I almost went there. Almost. Just by minute decision, I decided to go to Johns Hopkins instead. Also, he worked at the Howard Hughes Institute, one of the richest research supporting institute in the world, which is also situated near Johns Hopkins, and I knew the place. They've got lots of money, so if you want money, you can ask for contribution from them. So today, as we listen to his insights, let's reflect on our responsibilities and opportunities as educators, researchers, and policymakers to effect meaningful change together. I believe that we can employ the collective strength of our institutions, communities, and nation to address our contemporary urgencies. I appreciate very much your commitment to this noble endeavor, and I eagerly anticipate the discussions and outcome of this pivotal event. Thank you very much, ladies and gentlemen.
Thank you, Dr. Narengchai, for those insightful words.
ลำดับต่อไปนะครับขออนุญาตกราบเรียนเชิญมิสเตอร์อูเว มอราเวสประธานผู้ก่อตั้งมูลนิธิสันติภาพสากลขึ้นกล่าวความเป็นมาของการจัดงานในครั้งนี้ครับ Next, I would like to introduce Mr. Uwe Morawetz, the founding chairman of the International Peace Foundation.
So welcome to the Japan-ASEAN Bridges event series, which is facilitated by the Vienna-based International Peace Foundation. The events are hosted in cooperation with various local partners, including some of the country's major universities, and I would like to thank Khon Kaen University for hosting our event today. Commemorating the 50th anniversary of official relations between Japan and the ASEAN region, Bridges has been continuously held in Thailand and Japan since November last year, involving the participation of Nobel Laureates for physics, chemistry, medicine, and economics. The Japan-ASEAN Bridges series follows the series of over 800 Bridges events, which the International Peace Foundation has facilitated since 2003 to support education in the ASEAN region. The pluralistic program of Bridges highlights the International Peace Foundation's intercultural and transdisciplinary approach towards peace. The foundation doesn't take sides, but acts as a mediator by creating an independent platform for dialogue where representatives of science, politics, economy, culture, religion, the media, and youth can meet, share their viewpoints, listen to each other, and find mutual ways of understanding and cooperation. The aim of Bridges is to facilitate and strengthen dialogue and communication between societies in Asia with their multiple cultures and faiths, as well as with countries in other parts of the world to promote understanding and trust. The event's aim at building bridges with local universities in Asia to establish long-term relationships with Nobel laureates in all fields, which result in common research programs and other forms of collaboration. By enhancing science, technology, and education as a basis for peace and development, the events lead to a better cooperation for the advancement of peace, freedom, and security in the region, with the active involvement of the young generation, Asia's key to the future. This is why Bridges is not designed as a one-time event, but as a continuing process of synergies of now over 2 decades to make the series of events a sustainable success for Thailand and for Asia as a whole. I'm grateful to our honorary chairman, Anant Panirachun, and to our partners and sponsors who have enabled us to make the idea of Bridges a reality. I would like to thank everyone present today for taking part in this program. May it help us to facilitate a new culture of peace through dialogue, transcending its definition as merely the absence of war or armed conflict, into a new understanding what the basis for peace is: education. In this spirit, we welcome today the 2013 Nobel Laureate for Medicine, Professor Randy Schekman, who has agreed to come to Thailand to support the events. We all look forward to his keynote speech and to his important contribution to build bridges. A warm welcome, Professor Schekman. Khop kun kap.
Thank you. Thank you, Mr. Uwe Morawetz, for those fine remarks.
Next, it's time for a token of appreciation. May I invite Dr. Narongchai and Professor Schekman on the stage. Dr. Narongchai will be presenting a token of appreciation to Professor Schekman. There is an exchange of token of appreciation as well.
Yes, thank you, Mr. Chairman, for this warm gift. I have my own gift to give you, so please join me on this occasion of the 60th anniversary of the university and this distinguished audience and all of our guests. I want to present you with your own chocolate Nobel medal.
I have a Nobel Prize without doing anything.
Yeah.
Thank you very much.
It's not gold, but it is chocolate. But I wouldn't recommend eating it because the chocolate may be stale at this point.
Thank you. It was such a sweet moment. Next, I would like to also invite Associate Professor Shanchai and Mr. Morawetz to the stage, please. Associate Professor Shanchai will be presenting KKU's token of appreciation to the International Peace Foundation.
And Mr. President, I have for you your own chocolate Nobel Prize.
Thank you.
Professor Randy, you're starting something here. You better have a truck of chocolate medals in the back.
And it is time for photo taking. So could you please stay in line? Okay. Okay, and next it is time for group photo. So let me invite all the distinguished guests for today. Besides the most important 4 people on the stage, I would like to invite Mr. Surapon Pedwara, the Vice Chairman of KKU University Council, Honorary Consul of France at Khon Kaen, and Vice Presidents of Khon Kaen University, all the KKU Council members, Deans, Assistant Vice Presidents, Vice Deans, and Mr. Shanchai Sonti.
This is the logistical part of the event.
Can I count 1, 2, 3, and everyone cheese? Okay, 1, 2, 3, cheese. All right. Thank you very much, all the distinguished guests. Next, we are going to have the last photo session, but this session is going to be a selfie with all the audiences. So the cameraman will be on the stage and then take pictures with the audiences today. Okay, the first pose will be sitting pose. Okay. Let it be like an informal pose. How about that? Informal, freestyle. Okay, thumbs up pose. All right, next, it is going to be a standing pose. Please, everyone, stand up. Wake up.
Wake up, students.
Wake up. All right, let's make a thumbs up pose once again. How about thumbs up pose? Okay, like this. Oh, okay. We want a 60 years anniversary pose for KKU. How can you do like 6 and 0? 60. The president shows us like this. 6 and then 0. 60 years anniversary. Okay, ready? All right. Thank you very much. Thank you, thank you, thank you.
For those who weren't sure in what country you were, this is a land of pictures, right? Thailand loves pictures, so we do that. All right. As the podium gets back, now the moment we've been waiting for, I'd like to welcome Professor Randy Schekman, the 2013 Nobel Prize laureate in Physiology and Medicine, and who hails from the great Department of Molecular and Cell Biology Department at the University of California, Berkeley. Professor Schekman will present what you know already, the dialogue on the role of public universities as an engine for social change. Professor Randy, welcome. This is all for you.
Welcome, Professor Randy.
Let me get this going here. All right, now I can't, the cursor doesn't work. I'm going to have to unplug this to get the cursor. This pointer doesn't work. Okay. Okay, let's do this.
Sorry. Ready.
Okay, step aside here. So, good afternoon. Now that you're all energized and exercised, I want to tell you for the next 40 minutes or so about my journey and the journey of another scientist. Both of us relied on public higher education for our achievements. I grew up in Southern California, where my family was a middle-class family. There were 5 children in my family. There wasn't enough money to send us to a selective private university. Public education was what gave me a leg up and gave me the opportunity to do what I'm going to tell you about this afternoon in the scientific world. So let me tell you first about another example of a brilliant scientist, a Chinese-American scientist by the name of Roger Tsien. So I'll go to my slides now. Roger Tsien grew up in New Jersey. His parents and his grandparents immigrated from mainland China, where the family was a very highly accomplished family of engineers and scientists. So Roger had the benefit of that exposure from a very early age. He developed an interest in chemistry and physics, and in his school years, finally in high school, he advanced to such a level that he became the award winner of the most prestigious science competition in the USA for school children. It was called the Westinghouse Science Talent Search, and here is young Roger in high school with his, the work that gave him this prize, which provided him with a scholarship that allowed him to go to Harvard University, where he excelled in chemistry and physics. He went then to Cambridge University to do his PhD in physiology. And then, subsequently, he was recruited all over the US, but fortunately, we at the University of California at Berkeley were able to recruit him to be an assistant professor in my department. In his years, both at Berkeley and subsequently, he revolutionized our ability to look at molecules in living cells. He devised specialized fluorescent probes that allowed us to visualize the flow of calcium ions into cells and into compartments of cells while they were still living and responding to physiologic stimuli. He had an opportunity to move to the University of California, San Diego, where he was given a position as a Howard Hughes investigator. Howard Hughes supported his research for the rest of his life. In the early 1980s, a Japanese scientist at a laboratory in Massachusetts, the Woods Hole Marine Laboratory, discovered a protein in jellyfish that had an intrinsic ability to fluoresce. This protein was purified by Professor Shimamura, and he called it the green fluorescent protein. It wasn't clear what the protein did, but unlike any other protein molecule that had ever been described, this protein had an intrinsic ability to fluoresce. The structure of the protein created an apolar hydrophobic interior into which 3 amino acid tryptophan side chains were projected in an electronic configuration that allowed the molecule to absorb ultraviolet light and to emit in a visible range a green, yellowish hued color. This was entirely due to amino acids and not to some other organic fluorescent molecule. So that was a curiosity at the time. But in the years after that, another scientist at the Woods Hole Laboratory was able to isolate the gene from jellyfish, from the jellyfish genome, that encoded this protein. And Roger realized that when the gene was available, that it would be an amazing tool to allow one to tag proteins in living cells and to watch as they move around in the cell, dragging along this green fluorescent protein. So he and another scientist at Columbia University, Martin Chalfie, published important papers in the early 1980s where they showed that if you take the gene for this jellyfish protein and you stitch it next to a gene of interest, a human gene, any gene that you wanted to study, you would create a hybrid protein where your protein of interest was tagged with an intrinsically fluorescent molecule. You could then take this combined gene and introduce it into a cell, a human cell growing in the laboratory, or even into an organism. And then in real time, in living cells, you could watch as the green fluorescent probe followed the molecule that you were studying of interest as it moved around in the cell. This discovery and application changed the complexion of modern cell biology. It has allowed, in the years since these discoveries, the possibility to study living cells and to see how molecules move around. But Roger took this one step further. He realized that in the structure of this protein, in the apolar interior, there were other amino acids that bordered this tryptophan residue that influenced the emission spectrum of the molecule. He further reasoned that it might be possible to produce mutations in different amino acid positions that might change the electronic configuration of those tryptophan residues, thus causing the protein to absorb ultraviolet light, but then possibly to emit it in a different wavelength than the original green fluorescent protein. Now, he did this very systematically, changing the amino acids in the interior of this molecule to create an amazing rainbow of different molecules, each of which is based on the original green fluorescent protein, but with mutations that cause the emission spectrum to change to any one of these individual protein molecules. Now, not only is this an amazing result and colorful and beautiful, but the practical application of this was it allowed an experimentalist to take any number of these different fluorescent molecules and tag different other proteins in the cell, and to be able, with optical filters, to observe many different proteins as they move around the cell simultaneously, observing the emission spectrum with different color filters. This, too, changed the manner in which we conduct science at the cellular level. One simple example is shown here. This is a cell grown in the laboratory that is caught in the act of cell division. These fibers, labeled with one of the GFP variants, whose emission is orange, represent filaments of a cytoskeletal protein called actin. Blue was a different probe to label chromosomes, and green was yet another version, GFP, that labels another element called the microtubule, and this structure is the spindle on which chromosomes align to allow, during cell division, the sister chromosomes to be separated from one another. We now have these tools as a result of Roger's genius, and it has changed the way that we as scientists can investigate proteins of any nature in almost any cell, indeed in whole organisms. It's possible to visualize a developing embryo and to observe many different molecules in all of the cells of the embryo as it grows and matures. Well, for these discoveries, Roger received the Nobel Prize and continued his brilliant pioneering work in San Diego. Regrettably, all too young, he suffered a stroke and died prematurely in his early 60s, a great loss to the scientific community and to the world. But I want to leave you just with one final message about Roger. All of these achievements that I've just described were done at public institutions where the investment, in this case, of the citizens of the state of California made his discoveries possible. Well, I too was a beneficiary of that investment. I grew up in Southern California in an area called Orange County, but it was long before it was as heavily populated as it is now. There was a river running near my home, and it collected water into a still pond that grew some flora and a layer of pond scum on top of this pond. And one day, for reasons that I don't really understand, I took a jar and I went to this pond and I collected some dirty water. And I, for my 11th or 12th birthday, had received a toy microscope. And I took some of this pond water and I put it on a slide. And I couldn't believe what I could see even with this toy. I could see a world of complexity greater than what I see right now of microorganisms swimming and crawling and bumping into each other, eating each other. It was an amazing, amazing display of life at the microscopic level. One evening at dinner, I was expressing my enthusiasm for what I'd seen, and my father was rather skeptical. He couldn't believe that I could see these things in a toy microscope. So I decided at that point that I was going to get a serious microscope, a student professional microscope. So I used to babysit neighborhood children, I used to deliver newspapers, I used to mow lawns, and I collected my savings in an envelope that I put in the closet in my bedroom, with the goal in 1962 to save $100 US to buy a real microscope. Unfortunately, my mother kept borrowing the money to buy groceries, and I could never get the $100 that I needed. So one Saturday morning, I was so angry that when I finished mowing a neighbor's lawn, I rode my bicycle to a local police station, and I told the duty officer that I was running away from home because my mother was stealing my money and I couldn't get my microscope. So they called my father in, and he went into the captain's office, and there were some words behind a closed door, and he came out quite angry, obviously. But I learned a valuable lesson, because that afternoon, the $100 mysteriously appeared, and we rode to a local secondhand store, and I bought this Bausch & Lomb student professional microscope, which became my pride and joy throughout high school. Every year, I joined my fellow students in a science fair at the local school, at the county, and eventually in the state of California, and we mounted science fair projects for others to see, where we had the opportunity to develop our own interests and to follow our own instincts, reading what papers or books we could read to mount a little project of some interest. This alone was what stimulated my interest in science. And when I got to university, of course, the microscope got put away because the university had better microscopes. And when I was at UCLA, where I was an undergraduate, I learned in my freshman chemistry class about the nature of experimental science in the university, a really serious increase in the intensity of the work compared to what I could do in high school. And so I developed an interest in chromosome replication, and I did well, so that I was able, as a graduate student, to study one of the great with one of the great biochemists of the 20th century, a man named Arthur Kornberg at Stanford University. But now I'm going to tell you about my journey after that, because in 1976, I had the opportunity to take a faculty position at UC Berkeley, where I have remained ever since, now going on 48 years. I chose for my own independent path to follow something new that hadn't really been understood at a molecular level, using the tools that I had learned as an undergraduate and then as a graduate student. So let me now take you on a journey into cells at a different level than you've just seen. We're going to look first at a cartoon of what a cell membrane looks like. So this is a representation of a biological membrane. Many of you who have had an introductory biology class will know that all membranes, those that surround the cell or that surround compartments within a cell, consist of a bilayer, 2 layers of molecules called phospholipids, here depicted in red. The phospholipid consists of a polar hydrophilic head group, these red dots, that face either the outside of the cell or face the internal cytoplasmic surface of the cell. But attached to these head groups are long-chain fatty acids. These are hydrophobic, apolar. They don't do well when faced with an aqueous environment. And so when you have lipids of this structure and you simply mix them in a test tube, in a buffer, they spontaneously form a bilayer where the head groups project to face the aqueous environment, and the fatty acids bury into the inside of the bilayer to provide an apolar environment in which they are satisfied. Now, if membranes were only these bilayers of phospholipids, they would be impermeant to charged species that cannot cross such a hydrophobic barrier. So therefore, cells over 3 billion years of life on Earth have evolved protein molecules, these green structures, that allow cells to communicate with the outside in various ways. They are molecules like channels or permeases that allow ions or other small molecules to be captured and concentrated in a cell or to be expelled from a cell. Or they represent receptor proteins in a membrane to which hormones, molecules like insulin, bind to signal some event to the cell to change some aspect of its metabolism. Now, cells, as you'll see in a moment, are very complicated. Most cells that have a nucleus have many membranes, not just the cell surface membrane. Here, for instance, is one of the most dramatic cells in our body. This is called the beta cell of the pancreas. This is the cell, the tissue in the pancreas that is responsible for manufacturing insulin, that is what we use to control the level of glucose in our body. Insulin is made inside the cytoplasm and is captured into little granules that are surrounded each by its own unique bilayer membrane. Insulin is one of thousands of examples of proteins that must be encapsulated and segregated from other cytoplasmic proteins to enable their export out of cells. The human genome codes for approximately 23,000 different protein molecules, each encoded by its own unique gene. All of those proteins are manufactured inside the cell. They take their instructions from the gene, and they are assembled by stitching together long chains of amino acids. Proteins, the catalysts of most chemistry in a cell, have 20 different amino acids. And in different combinations, these amino acids create a long polymer that will eventually fold into a compact tertiary structure to catalyze some structural or enzymatic function. All of those molecules are made inside the cell. But some molecules like insulin, indeed thousands of molecules like insulin, have to be exported out of the cell. They are made inside the cell, but then the cell has to have a way of shipping them outside the cell. So cells have evolved in 2 billion years an elaborate machinery that recognizes those protein molecules that have to be shipped outside of the cell to distinguish them from the rest of the molecules that have to stay inside the cell. Much of what we knew by the early 1970s about how this process works was established by a brilliant Romanian scientist who did his work in New York at the Rockefeller University. His name is George Palade, quite a brilliant scholar in many dimensions, a very cultured man that I had the privilege of meeting when I was a graduate student in Arthur Kornberg's lab. He is seated here beside his favorite instrument. This is an electron microscope. Now, electron microscopes were built initially by German engineers who realized that it would be possible to project a beam, an intense beam of electrons onto a surface of a material, and the scattering of the electrons would allow the structure, the surface structure of that material to be deduced at atomic resolution. So it became possible to use this instrument to visualize materials at a microscopic level. Of course, this intense beam of electrons would be too great to allow a tissue to be preserved, to be able to visualize cells. And so what Palade did over the course of about a decade in the 1950s and 60s was, with his team, he devised procedures that could be used to chemically fix tissue slices so that the molecules in the tissue were rigidly held together and wouldn't move at all. He then found that such fixed tissues could be subject to dehydration, where the water was replaced by an organic solvent, and then these solvent-infused tissues could be mixed with a plastic resin that would polymerize to create a solid block. This block could then be trimmed and eventually cut to thin ribbons with a very fine diamond knife. And these ribbons then can be placed in a vacuum chamber at the base of this electron beam, and the fixed samples will resist the electrons that penetrate through the section. The clear areas of cells, electrons will just flow right through, but the structures within cells, particles that may be stained with chemical dyes, would bounce the electrons, and so one would see projected below the screen an image of a kind of a shadow of the cell. And the image that I just showed a moment ago is precisely that kind of image. Well, this technique, in its day, revolutionized our ability to look at cells, in this case, of course, dead cells, but to study the internal architecture of all cells. And so this changed, at that time, the way that we could examine what was going on in a cell. One of the things that Palade focused on was how a molecule like insulin becomes encapsulated for its eventual export outside of the cell. An image that you see here of higher magnification than the one I showed a couple of minutes ago is the last step before insulin is ejected out of the cell. The granule that houses insulin, where it had been encapsulated by a process I'll describe later, eventually migrates up to the perimeter of the cell, and the membrane of the granule touches the cytoplasmic surface of the plasma membrane. And at a key moment that is triggered and regulated, these membranes merge by a process called membrane fusion. This results in the ejection of the internal content of the granule to the outside of the cell without the rupture of the cell at all. This crystalline form of insulin then dissolves, courses through your body, collects in various tissues where it binds to a receptor on a muscle or a fat cell, and it tells that cell to swallow up the glucose that you've just produced by digestion. That's how insulin works. This event, membrane fusion, resulting in the secretion of insulin, is repeated billions and billions of times in almost all of the cells in your body. Here's a very different example, but uses the same logic. In your brain, or at the junction between nerve cells and muscle cells. Small granules, in this case they're called synaptic vesicles, which carry not insulin, but rather chemical neurotransmitters, are produced and gather at the nerve terminal. Some of these synaptic vesicles touch and dock and remain docked on the cytoplasmic surface of the nerve terminal. And when a nerve cell is activated by a change in the potential, membrane potential across the plasma membrane, that membrane potential surrounds the cell, travels down to the nerve terminal, and at a key moment at the nerve terminal, channels that allow calcium ion to flow into the cell touch these docked vesicles and cause them within a microsecond to fuse very rapidly. Membrane fusion in this case, very tightly, rapidly controlled. That results in the release, not of insulin, but of chemical neurotransmitters. These are the chemicals that mediate the flow of information in your brain. They are the molecules like dopamine or serotonin or acetylcholine, major chemicals that mediate traffic of information in the brain. In this case, these chemicals, acetylcholine in particular, diffuse across a gap, the synapse between the nerve terminal and a muscle cell, and acetylcholine activates a channel in the muscle cell that eventually causes the muscle cell to contract. Every time you move, this is being coordinated by millions and millions of cells. And in your brain, this is going on at a pace that it's hard to comprehend. Your brain has tens of billions of nerve cells. Each nerve cell can make hundreds or up to 1 million different connections with other nerve cells. This massive network of nerve interactions involves a communication that is absolutely dependent on the controlled fusion of synaptic vesicles to result in the release locally of chemical neurotransmitters. That flow of information, that critical membrane fusion step, is what controls every thought process you have, every memory you have, every emotion you have. All of that is tightly and still largely not understood, but controlled by that membrane fusion event. So bear that in mind as I then return to the discoveries of Palade that allowed us to see how these molecules progress from deep inside the cell to the cell perimeter. In 1974, George Palade won the Nobel Prize, and just after his receipt of the prize in Stockholm in December of that year, he traveled to San Diego, where he gave a lecture at the annual meeting of the American Society for Cell Biology. I had just left my graduate work to go to a laboratory where I was learning about membranes at UC San Diego, and I had the pleasure of watching Dr. Palade deliver a version of his Nobel lecture to a very appreciative audience of cell biologists. Here is what he discovered. Palade, studying the pancreas, was able to show that molecules like insulin are synthesized by ribosomes. Ribosomes are the little kind of sewing machines that stitch amino acids next to one another. Those ribosomes in a pancreatic cell are mostly lodged on the cytoplasmic surface of an internal network of membranes called the endoplasmic reticulum, or ER. He reasoned that these ribosomes must make polypeptides that thread across a bilayer of the ER to fold and form their final structure within the interior space, this salmon-colored interior space, which is called the lumen of the ER. He was able, using techniques that I don't have time to describe, to observe, largely in dead cells, that some of these proteins that are assembled into this canal-like network are captured into small carriers, other kinds of vesicles, that convey their cargo molecules through another organelle called the Golgi apparatus. The Golgi apparatus had been visualized by an Italian scientist by the name of Camillo Golgi in the late 19th century. It was seen in all cells, but it wasn't clear what it was doing. What Palade was able to conclude was this was a kind of a station along the way of a path that led from the endoplasmic reticulum eventually to the cell surface. At the Golgi apparatus, granules, including those that house insulin, are formed by a pinching process, and the granules are then directed to the cell surface where they finally discharge their content by this process of membrane fusion. So this was a beautiful demonstration of an elaborate pathway that had evolved over 2 billion years of evolution. And yet, in 1974, when Palade described his crowning achievement, we did not know anything about how this process worked. We knew the stages in the process, but we knew not a single molecule, not a single protein or lipid that had some role, some functional role. We didn't know the nature of the channel through which these polypeptides must pass. We didn't know how molecules were selectively captured and into vesicles that pinch from one another stage in the pathway. And we did not know what catalyzed this process of fusion, so vital in the pancreatic beta cell and indeed in almost all the cells in your body. So this was a challenge that I decided to take for myself when I had the opportunity to begin my laboratory at the University of California at Berkeley. Now at that time, when I started, 1976, the cell biologists who were the pioneers of Palade's generation were studying animals or animal cells or human cells grown in the laboratory. That seemed like the obvious thing to study to try to understand this process. But in 1976, the tools to study animals or animal cells grown in culture were very primitive. It would be difficult to use the tools of molecular biology by isolating mutations and isolating genes in animal cells at that time. In contrast, an emerging discipline in cell biology and molecular biology was the use of a very simple single-celled organism called Baker's yeast. You all know about Baker's yeast, the practical applications of Baker's yeast, but you may not know that as a tool to study cell biology, it had unlimited potential to reveal the nature of the mechanisms of essential processes. These are simple cells that can be grown in large quantities in the laboratory. They have a very simple growth style. At the beginning of a cell division cycle, under the right conditions of nutrition, a yeast cell starts to make a bud. The bud grows in preference to the mother cell for the next 90 minutes or so, until the bud is the same size as the mother cell, in which case the cell divides by a budding mechanism, and this can be repeated over and over again to produce billions of identical copies of the cells. Now, it has the virtue of a simple growth medium and absolutely identical cells, but most importantly, it's possible to use a simple form of traditional genetics to discover the genes that are required for all the processes that make a cell grow and divide. So we reasoned that it would be possible to use genetics and then biochemistry, because you could produce so many cells, to understand this Palade pathway. So the way to do this was to expose yeast cells to a chemical that causes mutations, and then to ask for those cells that are defective in the process of secretion. But let me show you first what a yeast cell looks like in contrast to a pancreatic beta cell. So here's a yeast cell, a thin slice, examined in the electron microscope. It doesn't look nearly as exciting as a pancreatic cell, but all the machinery is there. Most of it is a densely packed collection of ribosomes. The background granular staining in the cytoplasm of a yeast cell are all the thousands of ribosomes that are producing the 6,000 or so different protein molecules that a yeast cell needs to grow and divide. On close inspection, of course, you can see that there are membranes, There's a structure that is the equivalent of the lysosome in human cells. It's called the vacuole. There are membranes that can be seen here that touch the plasma membrane. In this case, these are the ER membranes of yeast. In other slices through other dimensions of the cell, you can see a nucleus with 2 membranes, just really very much like a mammalian cell. But our attention, and I'll show you the student responsible for this work, was drawn to a collection of small vesicles. These look, very much like the synaptic vesicles that I showed you a moment ago, but of course, yeast cells are not nerve cells. They are not secreting insulin. They are not secreting chemical neurotransmitters, but they are producing and secreting the molecules that a yeast cell needs to grow, on the surface of a grape, for instance. There are enzymes that a yeast cell secretes that are captured into these vesicles, which are discharged by this membrane fusion process at the surface of the bud. Now, Peter Novick, one of my first graduate students, shown here, busy pipetting away, Peter Novick and I imagined that these vesicles, shown in this slice, may be responsible not only for secretion, but also would be building blocks for the growth of the plasma membrane. Thus, when a vesicle touches and then merges by fusion at the cell perimeter, the membrane of the vesicle would become inserted into the membrane of the plasma membrane, so it would grow as each fusion event occurred. So that simple hypothesis led to a powerful prediction. The prediction is that if you cripple a gene required for the manufacture, for the traffic, or the fusion of these vesicles, that crippling event by mutation would kill the cell. And it's not much good working with dead cells, so you need to have a way of capturing such a lethal event under conditions where the cell can still survive. Fortunately, microbiologists had already figured this out long ago. It's very simple. You can do this with yeast. You can do this with bacteria. What you do is you take a culture, in this case of yeast, you expose it to a chemical that causes mutations, and the mutations spread out among the genome randomly. Occasionally, a mutation will change an amino acid in a protein molecule that will cause that protein molecule to be unstable. Sometimes that instability is reflected in a thermal instability, where the protein works normally, let's say at room temperature, but when the cell is warmed up to human body temperature, that mutation may cause the protein to unfold, to lose its function. There are many such mutations that can be introduced randomly and used to identify essential genes. These essential genes, if mutated in this way, will allow a cell to grow on a petri plate and form a colony at room temperature. But if warmed to 37 degrees centigrade, those cells in that colony will no longer grow. This is really a trivial way of identifying the essential genes in the yeast genome. I told you that yeast cells manufacture about 6,000 different proteins. Among them, 1,500 are known to be essential. That is, if you delete any one of those genes, the cell will simply die. But if you mutate them selectively, you may be able to produce forms of the essential protein that are thermally unstable. So that's what we did. We took a collection of randomly assorted temperature-sensitive yeast colonies, and Peter, as shown here, pipetting away, looked among them for mutants that cause enzymes that normally get secreted outside of the cell to build up inside the cell. That would distinguish such mutations from other mutations that cripple chromosome replication or gene expression. It was a unique phenotype that we could score. Well, the first such mutation we found was near the end of near the beginning of 1978. And at that time, Novick, having found this mutant, we had the opportunity to have Dr. George Palade visit Berkeley for an honorific lecture, and I was pleased to be able to tell him about our work, but more importantly, the students sponsored a dinner for Dr. Palade that evening, and Peter told him about his preliminary results with the first such mutant, which we called SEC1, short for secretion 1. Pilates said, well, of course, now you must examine these cells by electron microscopy. And of course, we were going to do that. We raced ahead and did this. And then one day, in the middle of the afternoon, Peter called me in my office. He was down in the basement in the electron microscope room. Very excitedly, he said, come down and have a look at the SEC1 mutant. Now, in contrast to the cell that you see here, SEC1, when these cells are incubated at 37 degrees for a couple of hours, but before they die, they start to accumulate thousands of these vesicles, no longer restricted to the bud portion of the dividing cell. They fill up the entire cytoplasmic volume because the SEC1 protein, being defective in these mutants, prevents the vesicle from docking and fusing at the cell perimeter. So the vesicles continue to be made until the cell basically chokes itself to death. This was very exciting to us, and of course, over the next year and a half, we then repeated this procedure over and over and over again and collected hundreds of such mutations, and in time, we were able to chart a path that's shown here of genes such as SEC1, but now many more genes indicating many more proteins required at each of the several stages in this pathway. And over now decades later, 100 or more such genes are known in yeast, including the genes, for instance, that encode the channel in the ER membrane that initiates this process. Genes that control a bidirectional flow of vesicles back and forth between the ER and Golgi. Genes that control sorting of proteins to the lysosome-like structure, and so on. After we had all these mutants, it became possible to clone the genes and to sequence the genes and to discover what the predicted protein sequences were. But at the time, the genome databases had few entries, and there was nothing in the database that looked anything like the genes that we found. In time, the human genome was sequenced, and not surprisingly, all the yeast genes that we found are found in the human genome. Indeed, the SEC1 gene that starts this story is found expressed in your brain, and it controls the fusion of synaptic vesicles at the presynaptic membrane, virtually the same protein. And this emphasizes, again, the unity of biology. There is an evolutionary conservation. When nature evolves a solution to a complex problem of how to capture proteins for export from a cell, that machinery gets used over and over and over again. And I use this illustration of a cluster of yeast cells, which seems quite different than Charles Darwin, but the fundamental mechanisms that operate in the body of Charles Darwin and all of us are much the same, and they were evolved 2 billion years ago. Now, just in closing, at the time that we were making these discoveries, the biotechnology industry was growing up in the San Francisco Bay Area, and I had the opportunity to collaborate with a local company to help them exploit this discovery for the manufacture of clinically important proteins that would be difficult to isolate by other means. And so, for example, the gene for human insulin, which had been cloned, was introduced into a yeast cell, and now, many years later, 1/3 of the world's supply of human recombinant insulin that's used to treat diabetics is made in giant fermentation plants with growing yeast cultures. Similarly, this company took the gene for the virus, hepatitis B virus, surface antigen, and when that gene is put into a yeast cell, that antigen, which is a membrane protein, collects in little vesicles, and for any of you who have been vaccinated against hepatitis B, you've received little vesicles isolated from yeast cells. Well, let me bring this story full circle. Of course, you recall that microscope that started my journey in cells got put away. Fortunately, my parents saved that microscope. They sent it up to me, to my family near Berkeley. Regrettably, neither of my children ever showed any interest in science, So the microscope sat and collected dust over many years. And on the days after I learned of my Nobel Prize, I and my fellow laureates received an invitation from the Nobel Museum in Stockholm to send in some artifact that reflects a part of one's discovery. In my case, it didn't seem interesting to send in Petri plates, so I dusted off and sent in my microscope. And if you're ever in Stockholm, you can come by and see that microscope. That was what I used to begin my career, including in English and in Swedish, a caption of how I had to run away from home to begin my career in science. Thank you for your attention. Now I think we have time for questions. Do I organize this? Okay. Whatever you would like to ask. Maybe if there's a microphone that can be passed along around the audience. Don't be shy.
Sure. We will now move on. Thank you, Professor Schekman, for your special session and the insightful information that you've given us. And we would like to now move on to take questions from the audience. So if you're interested in asking Professor Schekman some questions, please raise your hands.
Here's one.
Can you raise your hand up high?
I would like.
It's on.
I would like to ask you: do you approach any problem when you start a laboratory in 90s?
Yes. So the question is, did I encounter problems? I certainly did. When I started in 1976, I had worked on bacteria. I had never worked on yeast. I'd worked on membranes, but I had never studied secretion. And I had never done genetic analysis. But I thought I had good ideas, and I wrote them down for a grant to the National Institutes of Health, and it was completely destroyed. The study section felt that I had accomplishments in the past, but that I had no relevant experience, nor did I have any preliminary results to support my hypotheses. So that was a valuable lesson. Fortunately, I was able to get other small grants for a couple of years until we discovered this mutant, and then the NIH was very generous, and I've been supported ever since. So what this illustrates to me, and it's still true, is that the system is very conservative, and it's resistant to new ideas and new approaches. But it isn't, if you don't have the courage to pursue your own ideas and to have confidence in your own abilities, new discoveries will never be made. So of course, it was disappointing, but I didn't change my ambition or intentions at all. I was able to pursue what I wanted, and ultimately, it worked out very well. So that's a lesson for you, too. There's a question. Microphone up here, please.
So in your opinions, do you think that science research are more important than other fields of the study in terms of social changes?
In terms of?
Social changes.
No, I don't put science ahead of other intellectual or cultural endeavors. It just was what I found most satisfying in my life. I started university with the intention of going to medical school to become a physician, because that seemed to be the most logical application of what I was interested in high school. But I realized when I got to university in my freshman year that there was this world of science that I, of course, knew, but didn't realize that I would have the ability to enter a field of academic science. And so I had the good fortune of beginning to be able to work in a research laboratory at UCLA in my first year in university. And so that really changed my life. I went home to tell my parents that I wasn't going to be a real doctor after all. They were disappointed, but they got over it.
So another question. So as an international affairs student, do you have any advice from like adapting the scientific methods way to use as with the social way?
To use in which way?
With sociology.
Yes. Well, the social problems are vast and much more complicated than a simple microorganism. But there is fundamentally the logic of looking at a complex problem and seeing how you can solve it, and just being focused and persistent. I've felt in my own career, and I feel in many successful scientists, they have a passion to make a discovery in a certain area, and they focus their attention laser sharp on that one problem, turning the problem around in their head over and over until they find an angle to solve the problem. And I think that kind of intense focus and devotion can solve almost any problem. And let me give you another larger example of something that I've become involved in. So I had the misfortune of losing my wife to Parkinson's disease some 6.5 years ago. She suffered for about 20 years. And as she was in decline, and then when she died, I was given an opportunity to create a large international network of basic scientists studying Parkinson's disease. Parkinson's has been around, of course, since the beginning since man evolved, but it's been known as a disease for 200 years. And yet, in that 200 years, there are no cures. There are treatments that reduce the symptoms, but the disease inexorably progresses to death. But there are many clues, many genes that are now known in familial forms of the disease. So I felt, given my own personal experience, that this was something that this approach of intense focus and scientific curiosity is something that really needed to be mobilized on a worldwide scale, mobilized with a laser-sharp focus on Parkinson's, finding the molecules in the cells that lead to the initiation and progression of the disease. So now, 5 years into this program, we have 35 teams consisting of 163 laboratories in 80 different institutions and 14 countries around the world, all collaborating in real time with each other with experiments that are focused on molecular and cellular aspects of the disease. And I feel that focus and intense interest and interaction will make breakthroughs that have not yet been possible. So I think for a complicated problem like a disease that's been with us for so long, that same approach may prove beneficial, and I'm grateful to the benefactor, the billionaire who's been funding all of this work, making it possible. And I think the same logic could be applied to other complicated problems.
Thank you, sir.
Sure.
Any other questions?
Oh, we're getting more hands up now.
How does it really feel to be at the forefront of microbiology? I could not imagine how you feel to discover such things as how the vesicle would work inside of a cell, or how the graph endoplasmic reticulum worked.
Sure.
In these day and age, we can see everything in a textbook.
Yeah.
But you, being as you are, and your colleagues, discover these things at that time. How do you really feel about it?
Well, thank you. That's a great question. Thank you very much. Well, you should appreciate that when you look at a textbook, as you have, there's an awful lot of work that went into those discoveries that seem so matter of fact. Every sentence in every textbook of cell biology takes hundreds of people, many years, to make the discoveries that lead to the words in that sentence. So the feeling, I can tell you, is exhilarating. There are moments in my life when I've had that kind of what we call in English the eureka moment, where you know in your heart that something has just changed your life. And the moment that I shared with you, when Peter Novick called me down to the electron microscope room and I saw the image that I shared with you, I knew at that moment that I would have 20 years of exhilarating effort ahead to figure out this process that we had cracked through using genetics. And it took even more than 20 years, and I'm really grateful for having had that opportunity. I hope you too can have such an opportunity.
Thank you very much.
Any more questions?
There's another hand nearby. There you go.
Oh, I wanted to ask, you mentioned this cell, green fluorescent cell. It glows, right?
I'm sorry, I missed your question. Speak again.
This cell you mentioned, this green fluorescent cell.
Yes. Mm-hmm.
Does it found in other organisms other than jellyfish?
Yes, yes, yes. Very good question. Yes. So Shimomura's discovery was the first of many discoveries of other intrinsically fluorescent protein molecules isolated from a variety of different organisms. Some fluoresce emit in blue, some in red. So there are natural proteins. You don't have to have mutations. There are natural proteins that can be used in the same way that that I described, that Roger Chen used.
Does it work the same way as like in a firefly in a molecular basis?
Yeah.
Because it
firefly
glows.
is a different kind of chemical reaction. So firefly produce an enzyme called luciferase. Luciferases emit luminescence. It's not that they absorb ultraviolet light. They emit luminescent light, very often by using an energy-containing molecule like ATP. It's a different kind of chemical reaction, but equally dramatic.
So if you put this green fluorescent cell in like an organism with transparent skin, will it glow?
I'm sorry. Will it
glow like?
Flow. You can visualize now with special microscopes. You can visualize intact organisms, particularly organisms like zebrafish that are clear, that you can see right through. You can visualize many different molecules moving around in a living organism or in a developing embryo in real time. It's now possible using a very sophisticated so-called super resolution microscopy to visualize individual molecules, not just collections of molecules, individual molecules as they move around in a cell. So our ability to look at life in real time has exploded in the recent decades. Okay. There's a question for Mr. Chairman in the front. Winner of the Chocolate Nobel.
Thank you. I've been listening to you, smiling all the way. It seems to me that people like you will continue to discover how to live longer and longer. All kinds of cures are going to be invented. So I think, believing in what you said, I think we are entering into the age of longevity. Mm-hmm. I'm concerned about this age of longevity because society will change. Yeah. Can you share with us your views about the future? Yeah. When people don't die. Yeah. How do we manage when people just don't die? Yeah. Thank you.
That to me is a dystopian view of the world. I don't welcome that prospect. What we should seek in medical research is to extend the healthy lifespan, the healthy but normal lifespan. I believe in turnover in the world. I believe that people like Donald Trump should not live forever. So we need new young people who come in with new ideas and new vigor and new energy to bring new ideas to the front. Regrettably, as you see in the United States, we have 2 old men running for president. This is not good. We need younger people to take their place. So I think the goal of medical science should be to conquer the diseases that degrade the quality of life, but not necessarily extend the lifespan beyond a natural range. Of course, we all want to live a healthy life. Maybe some people want to live forever, but I don't think that's a good thing. And I think you probably share that view. Here's another question.
Good afternoon. I'm Celine, the Dean of KKUIC. So I'm from the social science side, not the biologist. My question is, how do you envision biology and social science collaborating to address ethical and policy implications of scientific advancement?
Great question, thank you. So scientists like me can do certain things, but we shouldn't be asked to do everything. Once the discoveries are made, their application or control needs to be decided by other people, social scientists, politicians. So in the current revolution in genome editing, where my great colleague Jennifer Doudna made a key discovery that led to her Nobel Prize, that technology has enormous power, but potential dangers. We worry about using that technology to manipulate human embryos. You can imagine good things that could be done to change the human embryo to make people who are smarter or who don't go bald or, or are great athletes. But you can also imagine other things that you wouldn't wish to see done by such manipulation. This cannot be determined by scientists, at least not alone. This must be people who think more broadly about science, about society and the interactions of people in society. So these decisions need to be made by wise leaders who take the information and use it or control it. We saw this in a dramatic form in the recent movie Oppenheimer, which you may have seen. I mean, here was an amazing discovery, an exhilarating manner in which the discovery was made, but the application of that dangerous knowledge really eventually made many of the scientists who participated wonder whether they were doing the right thing. But once done, it was unleashed, and then it was up to people, politicians, to decide how it may be used or not used. These are decisions that can't be left to scientists.
So if there's no more questions, please, a great round of applause for Professor Schekman. Thank you so much.
Thank you so much.
Professor Schekman will meet with academics and researchers outside of the room to talk about possible collaborations in the future. And I hand off the closing words to Charis.
And this concludes our event. We would like to thank all participants and everyone involved in making today's a success. We appreciate your engagement and contribution to this event. Thank you very, very much. And I would like all the students to remain. There will be an organizer to tell you about the evaluation form. Thank you very much.